Short questions and answers on enzyme inhibition. 1. What is enzyme inhibition? Enzyme inhibition is the phenomenon in which an enzyme fails to catalyze a reaction. 2. What are enzyme inhibitors? Molecules that react with an enzyme but are not converted into the desired products are called enzyme inhibitors. 3. Give examples of enzyme inhibitors. Examples include poisons, cyanides, antibodies, anti-metabolites, penicillin, and sulfur drugs. 4. What are the two types of enzyme inhibition? Enzyme inhibition can be competitive or non-competitive. 5. What is competitive inhibition? Competitive inhibition occurs when a chemical competes with the substrate for binding to the enzyme's active site, blocking enzyme activity. 6. What is a competitive inhibitor? A competitive inhibitor is structurally similar to the substrate and fits into the enzyme's active site but cannot be catalyzed into products. 7. Is competitive inhibition reversible or irreversible? Competitive inhibition is reversible. 8. What determines the level of competitive inhibition? The level of inhibition depends on the relative concentrations of substrate and inhibitor. 9. Give an example of a competitive inhibitor. Malonate is a competitive inhibitor of succinate dehydrogenase. 10. What reaction is catalyzed by succinate dehydrogenase? Succinate dehydrogenase catalyzes the conversion of succinate to fumarate. 11. Why are competitive inhibitors important? A. They support the lock and key hypothesis. B. They show that structurally similar substances to substrates are not acted upon by enzymes. C. They are used as drugs, such as sulfonamides, to control bacterial pathogens. 12. What is non-competitive inhibition? Non-competitive inhibition occurs when the inhibitor binds to a site other than the enzyme's active site, allosteric site. 13. What is the allosteric site? The allosteric site is a site on the enzyme where non-competitive inhibitors bind. 14. How does a reversible non-competitive inhibitor work? It prevents the formation of enzyme product complexes but does not stop the formation of enzyme substrate complexes. 15. Give an example of reversible non-competitive inhibition. Feedback inhibition is an example of reversible non-competitive inhibition. 16. What happens in irreversible non-competitive inhibition? The enzyme is permanently inactivated by altering its shape, making it unable to bind to the substrate. 17. Give examples of irreversible non-competitive inhibitors. Cyanides and salts of heavy metals such as mercury, silver, and copper. 18. How do cyanides act as inhibitors? Cyanides inhibit cytochrome oxidase by combining with iron in its prosthetic group, blocking cellular respiration. 19. How do heavy metals like mercury act as inhibitors? Heavy metal ions break disulfide bridges by combining with thiol groups in enzymes, denaturing them. 20. What is feedback inhibition? Feedback inhibition occurs when the activity of an enzyme is inhibited by its own product. 21. Is feedback inhibition reversible or irreversible? Feedback inhibition is a reversible non-competitive inhibition. 22. What is the role of feedback inhibition in cells? It regulates metabolic pathways by controlling enzyme activity. 23. Give an example of feedback inhibition. In the conversion of aspartate to threonine, excess threonine binds to an allosteric site on the first enzyme, inhibiting the pathway. 24. What happens to feedback inhibition when the product is consumed? When the product, example threonine, is consumed, the inhibitor detaches from the allosteric site, and enzyme activity resumes. Thanks for watching.